Hi students, today 11th classes. Already which topic is going on this class, cell biology. That is 6th chapter. This chapter, which topic is going to classify today is commonly called as flagella. Flagella is nothing but it is a microscopic structure that helps in locomotion. Flagella that are generally present in both prokaryotic organism as well as eukaryotic organism. In the class, which one is first going to discuss about is namely called as prokaryotic flagella. Prokaryotic flagella is commonly called as it is a helical appendages. What do you say? Prokaryotic flagella is commonly called as it is a helical appendages that help in motility. The prokaryotic flagella is commonly called as it is much thinner than the eukaryotic flagella because comparing eukaryotic, the flagella is a prokaryotic, it is a much thinner. In addition, that flagella, there are generally three components are there, namely basal body, hooks, then helical filament. According structural view, that is basal body. That's are commonly called as short hooks. The short hook which bear helical filament is commonly called as filament. There are three structural views that are present, namely basal body, hook and the filament. The basal body is generally attached to cell membrane. The basal body is always attached to cell wall. Hook is always attached to cell membrane. From hook, they can be producing extension projection is commonly called as a filament. The filament, which is made up of a special protein, is commonly called as flagellin. So that is generally present. Three basic structure is namely called as basal body, short hooks and the helical filaments. You know that flagella is a locomotory organ. For example, the bacteria which rotate that flagellum, the basal body that can be possessed, propel rings are there. That propel rings, that it will be act as rotatory motor. to say the basal body which are possess propel rings. That propel rings that are involved into rotatory motor. That rotatory motor spin the flagellum is become to rotate. These are things it is commonly present the structure of flagella. Next I am going to discuss about structure of flagellum in bacteria. Generally, there are two kinds of bacteria, you know that. Namely, one is gram-positive bacteria. Second one is gram-negative bacteria. How you know about the names must becomes under gram-positive bacteria. So which bacteria that are retain the purple color, those bacteria it must becomes under gram-positive bacteria. Now come down. The gram-positive bacteria that contain only two basal rings. Namely, S rings and M rings. What is the meaning commonly called as S ring? Super membrane ring. What is the M ring? Membrane ring. Now come to R. The S ring is always attached to inner peptidoglycan. The M ring is always attached to cell membrane. So there are two rings which are present to a gram-positive bacteria. Now comes on down. What is gram-negative bacteria? You know that I am already indicating to you. Which bacteria will not retain that purple color? Those are must become the gram-negative bacteria. The gram-negative bacteria they can be possessed. Two pairs of rings are there. The first ring is commonly called as, the two pairs of rings can be classified as two types. Namely, one is called proximal rings. Second one is distal rings. So you know that for the structural view, this is commonly called as cell membrane. This is commonly called as cell wall. So for here, this is S ring. This is M ring. This is L ring. This is A ring. So these two is commonly called as distal ring. These two is commonly called as proximal ring. So the proximal ring, distal ring, who responsible for touch is commonly called as central rod. I come to the point. For the proximal ring, they can be classified two types, namely L ring and P ring. What a L ring is made up of lipopolysaccharide. What is the P ring? Peptidoglycan ring. So now come to the point. Proximal ring, that's are situated to, or that's two is attached to the cell wall. The distal ring, that's called S ring, that's a super membrane ring, and membrane ring usually that must be attached to the cell membrane. Next one I am going to discuss about mechanism of flagellar movement. For the mechanism of flagellar movement, which be responsible for move with the help of protein, or also called proton. That to say is commonly called as proton motive force movement. I am already indicating to you, for the structure of flagella there can be possess four basal rings that are situated to the basal body. What a mechanism of flagellar movement, or also called proton motive force movement. In general, the flagellar movement, it is based on proton movements. So the flagellum is swim, the flagellum is a rotate based on protons movements that must be occur into the flagellum. In general, proton only it is involved to movement is takes place, but not involved in ATP. So without involvement of ATP, what is called ATP adenosine triphosphate, that is an energetic compound. Without the energetic compound, the flagellum motion is that takes place with the help of proton movements. Here, then the flagellum, all the protons flow back on the cell, or all the protons it move into the cell, where the proton it must move from in through basal body. So all the proton it must be reach on the basal body. In each flagella that must be swim, that must be drive, that must be rotated. So with the help of proton movement, the flagella to be swim, or it must be to rotate. So I am already indicating to you, there are four rings, So that flagellum, it must be moved with the help of proton, not in the ATP. So that here, the protons force are flowing back into the cell through the basal body, because the basal body that can be possessed of 4 rings, that's helping driven to rotate. So these rings are commonly called as rotatory motor. What is it? The basal rings or propel rings that are commonly called as rotatory motor. Now come to the point. What do you mean commonly called as proton motive force? Proton motive force that helps in driven to flagellar movements. So without force cannot be motion or cannot be bent to help in modality. So before entering like this, what do you mean commonly called as proton motive force? The force that is derived from electrical potential what is that? The proton motive force, the force is derived from electrical potential and hydrogen ion gradient across the plasma membrane or across the cytoplasmic membrane. So the proton motive force, the force is derived from electrical potentials and the hydrogen ion gradient across the cytoplasmic membrane. So the flagella, it must be rotate or it must be movements. The flagella, it's get from energy from proton gradient. So what is it? The flagella that must be moved to location. The flagella drive energy from proton gradients across that cell membrane generated by oxidative phosphorylation. What do you mean commonly called as oxidative phosphorylation? Phosphorylation which takes place during electron transport chain, that is commonly called as oxidative phosphorylation. Through oxidative phosphorylation, the flagella, it is receive energy from proton motive force or from proton gradient. So that the bacteria, the rotatory motor, that are generally situated to cell membrane. What is it? The bacteria, rotatory motor, that are present to cell membrane. Therefore, cell membrane is commonly called as, it is an actual site of generations of protein motive force. So that are the things commonly called as mechanism of flagellar movements. Next one I am going to discuss about. The early class to be discussing prokaryotic flagellum and the structure of flagella in bacteria and a mechanism of prokaryotic flagellum. Now the discussing topic is commonly called as eukaryotic flagellum. That's are commonly called as cell motility. The flagella is a microscopic structure that helps in locomotion. Likewise, the eukaryotic flagellum, that are developed from basal body. The structure of eukaryotic flagellum, it must be consist of So many kinds of compounds are there, namely they can be possess nine pairs of tubules. What to say? For the structures, it must be consist of one, two, three, four, five, six, seven, eight. There are nine pairs of microtubules are there. There are nine pairs of microtubules which are present to peripheral, that we say commonly called as peripheral microtubules. For that microtubules, there are two types are there. One is called tubules A, another one is called tubules B. These are called tubules B is commonly called as tubules A. Those two constitute is commonly called as doublets of microtubule, because two things. So there are nine pairs of peripheral microtubules are there. They can be classified as two types, namely tubules A and tubules B. Those are occur in a pair that we say called doublets microtubule. In there, two microtubules are there. So the outer nine tubules that are arranged look like circle or ring-like structure. So what the arrangement is commonly called as 9 plus 2 arrangements. So 9 pairs of peripheral microtubules and one pair or two microtubules which are present to center. In commonly, flagella is nothing but is a microtubular projection. What do you say? Flagella is nothing but it is a microtubular projection. In addition, flagella is a much longer than the cilia. So comparing cilia, the structure of flagella is a much longer. That longer is measured about 200 micrometer longer in flagella. The structure of flagella, it can be considered as, is commonly called as axoneme. What do you say? The structure of flagellum has an axonic, the second head. The axonic that must be considered as microtubules and protein tubulin. Next one I am going to discuss about movement of eukaryotic flagellum. What is that? Movement of eukaryotic flagellum. So I am already discussing about what is the structure which they may be present to be here. For that flagellum, 9 pairs of peripheral microtubules are there. That 9 pairs of peripheral microtubules, each pair possess axonin dynein. What is that? The peripheral microtubules having axonin dynein is a protein compound. What is the vital role of axonin dynein in peripheral? Each pair is commonly called as that are responsible for give force for movements. In eukaryotic flagellum, which way is responsible for movement is commonly called as each pair in peripheral tubules. They can be possess axonin dynein sheath or axonin dynein protein that's generative force to movements. That movement is generated by ATP. But a prokaryotic, the flagellum is responsible for move with the help of protein movement. It is not involved in ATP. But a eukaryotic organism, the flagella is responsible for move with the help of ATP. For the structural view, the interactions between tubulin and dynein. What is that? The interaction between tubulin and dynein. It is nothing but a mechanism of contractions in cilia and the flagella. What do you say? The interaction between tubulin and the dynein protein is the mechanism of. So those are things in combination that are responsible for contraction and relaxation. So which is responsible for going to be contraction is commonly called as tubulin and the dynein. They are responsible for contraction in cilia and the flagella. So for movement of flagella, that dynein which receive energy from ATP, that's derived energy from ATP. So these are things. So dynein is derived energy from ATP. That derived energies, it is shifted to adjacent microtubules. So what are the microtubules that must be present to adjacent to dynein sheath? That must be shifted. Due to the shifting nature, the flagella is going to movements. That's why the flagella cilia, it is a bent to happen in movements. So that to be say commonly called as eukaryotic flagellum cell mortality, as a commonly called as 9+2 arrangements. Outer peripheral 9 pairs of microtubules and inner one pair of microtubules. The tubules that must be connected to central spokes. That's called what that? Central spokes. These are called dynein proteins. So these are things which are present in the structure of movement of eukaryotic flagella. The next one topic is commonly called as cilia. Cilia is a plural form. In singular is commonly called as cilium. So plural form cilia, singular form cilium. So what do you mean cilia? Cilia is nothing but small, short, numerous projections in cell membrane. So comparing flagella, the cilia is a small, it is a short, it is a numerous number. It is occur in outer projections of cell membrane. For the cilium, it's a commonly called singular form. So what are the definitions of cilium is commonly called as? Cilium is commonly called as is a membrane bounded structure. It is membrane bounded structure. It includes cell body or rootlets, cell plate and shafts. Those things are there. What is the name they call as? Cilium is a membrane bounded structure. It includes cell body, rootlets, cell plate and shaft. Shaft is also named axoneme. Axoneme is a head, a structural view. The axoneme that must be considered as 9 pairs of microtubules that are situated towards periphery and 1 pairs of microtubules which are situated to center. That arrangement is how to call is commonly called as 9+2 arrangements. That lengths. So 1, 2, 3, 4, 5, 6, 7, 8, 9. These are 9 pairs. These are 1 pair. These are called 2. So the arrangement is commonly called as 9+2. For the axoneme, that must be considered as 9 pairs of microtubules towards the periphery and 1 pair of microtubules towards the center. The motor protein, that's a commonly called as called dynein. The motor protein dynein that's correct peripheral microtubules and central microtubules. So motor protein dynein that's connect peripheral microtubules and the central microtubules. Those things it responsible connect is commonly called as dynein. The one is called nexine. This is a called protein compound that are connect peripheral microtubules alone. These are commonly called as an exine. So the exine that responsible for connect peripheral microtubules. So these are things discussing today. Is nothing but prokaryotic flagellum and eukaryotic flagellum and cilia. Thank you.